Okay, before we fill in the contents of the inside of your foldable, we do want to name each of these phases, and you're eventually going to cut along the folds, the creases that you made earlier, so you can use these almost as flashcards. So for the names of our phases, our first phase, we'll write it and then turn this back so you can see it better, is interphase. Inter like in between. Then we have prophase, that's step 2. Metaphase, meta like middle. You can see from the picture where we're going with that one. Anaphase, telophase, and then lastly something called cytokinesis. Long word, but really we're talking about cytos and cell and kinesis like moving. These two cells are moving apart. So we're going to describe in words each of those phases. I'll give you a second to write them down. You had a template on haiku that I'm hoping you filled out already, so you can see that mine is filled out. If you did not do that, you'll want to go ahead and do that, and then we will complete the inside of this together. So starting in the upper right, which sounds a bit odd, upper left, sorry, it sounds a bit odd because it feels backwards on my screen right now to me, but here we go. This is describing interphase. It is the longest by far phase of the cell cycle. During this phase, the cell will grow a lot. Remember, cells can only reach a certain size before they die, so this is actually going to have a limit in how large the cell will be before it divides. It continues its normal functions. Things like respiration, photosynthesis, those are all happening. The DNA gets copied. That is known as DNA replication. So I'm just going to throw that word in there, replication, because we'll see that a lot in this unit. And it also copies the organelles. So for example, you may have noticed on the front that we had a centriole. Sorry, that's a little hard to see. A centriole in our first step, and then the second step there were 2 of them because it got copied. So that was interphase. Going over to step 2 now, this is prophase. Our chromatin condenses into chromosomes. So remember, we went from loose and thin chromatin to those X-shaped chromosomes. The spindle fibers form. And then the nuclear envelope, sometimes called the nuclear membrane, same thing, breaks down. Those are all happening in our picture on the front. Step 3, which was metaphase. Our chromosomes line up at the equator. I always think meta starts with M, so in meta they meet in the middle. The chromosomes attach to the spindle fibers at their centriole. So remember, they will attach right in the center of that X. Moving over, so we had interphase, prophase, metaphase, anaphase. In anaphase, I always think A means away. We're going to see our chromosomes actually separate here. So the centromere in the middle of the chromosome splits. The chromatids, each half of our X, separate and move to opposite sides. We call those opposite poles. Again, you can kind of think of the cell as a globe here. The cell also elongates. So in this step, you start to see the cell sometimes look more like an oval shape because it is starting to stretch out. After anaphase, we have telophase. In telophase, our chromosomes will start to unwind again. So again, they're going to go back to that chromatin form. New nuclear envelopes, notice there are 2 of them, will form around our new nuclei. So by the time this phase is done, you actually have 2 separate distinct nuclei in one cell that's stretching out, beginning to get further and further away from each other. And lastly, in cytokinesis, in this step, we just get the cell membrane in an animal cell pinching If this were a plant cell, we get a new cell while growing. This cell splits in two. We get two new what we call daughter cells with identical DNA. That's really important. We are making exact replicas that are half the size of the original. Because remember, now we're going to go back to the beginning. This is the cycle. And these daughter cells will be in interphase and they're growing. So just putting this all together one more time. Interphase, the DNA gets copied. So I have exact copies of everything. Prophase, the DNA condenses. My spindles start to form. My membrane breaks down. Metaphase, everything meets in the middle, so my chromosomes are lined up single file down the equator. Anaphase, A means away. My sister chromatids, each half of that X splits, and the spindle fibers pull them towards the poles of the cell. The cell gets a little bit longer. Telophase, it continues to elongate. I start to see the regeneration of these nuclear membranes. Eventually I will have two separate nuclei when I get to the end of this phase, and my DNA is unwinding. Spindle fibers are breaking down. There's a lot happening in prophase and telophase, but they're the opposite. So if you can remember what happened in prophase, just break it back down in telophase. Lastly, cytokinesis. We see the division of the cytoplasm has nothing to do with the nuclei anymore. We're actually splitting the cell itself. So at that point, we have two identical daughter cells.